Chapter 1. Reproduction in lower and higher plants. Reproduction is the production of young ones like parents. Reproduction is an essential process as it leads to continuation of species as well as to maintain the continuity of life. Each organism has its own particular method of reproduction. All these methods generally fall into two categories: asexual reproduction, sexual reproduction. 1.1. Asexual reproduction. Asexual reproduction does not involve fusion of two compatible gametes or sex cells. It is the process resulting in production of genetically identical progeny from a single organism and inherits the genes of parents. Such morphological and genetically identical individuals are called clones. Organisms choose to reproduce asexually by different modes or ways. First, fragmentation. Multicellular organisms can break into fragments due to one or the other reasons. Example, Spirogyra. These fragments grow into new individuals. Second, budding. It is the most common method of asexual reproduction in unicellular protozoan and yeast. Usually it takes place during favorable conditions by producing one or more outgrowths, buds. These buds on separation develop into new individual. Third, spore formation. In Chlamydomonas, asexual reproduction occurs by flagellated, motile zoospores which can grow independently into new individuals. Other methods of asexual reproduction include binary fission, which occurs in chlorella, diatoms, and Chlamydomonas, conidia formation in penicillium, and gamma formation in Marchantia. Vegetative reproduction. Plants reproduce asexually through the vegetative parts. Hence, the new plants formed are genetically identical to their parents. There are also few methods which would not occur naturally in the plants. Agriculture and horticulture exploit vegetative reproduction in order to multiply fresh stocks of plants. Artificial methods are used to propagate desired varieties according to human requirements. The various methods are as follows. A. Cutting. The small piece of any vegetative part of a plant having one or more bud is used for propagation, viz. Stem cutting, example rose, bougainvillea, leaf cutting, example sansevieria. Root cutting, example blackberry. B. Grafting. Here parts of two plants are joined in such a way that they grow as one plant. In this method, part of the stem containing more than one bud, cycon, is joined onto a rooted plant called stalk, is called grafting, whereas budding is also called bud grafting, in which only one bud is joined on the stalk. Example, apple, pear, rose, etc. Tissue culture. It is a method by which a small amount of plant tissue is carefully grown to give many plantlets. Micropropagation method is used nowadays. 1.2. Sexual reproduction. It involves fusion of two compatible gametes or sex cells. All organisms reach to maturity in their life before they can reproduce sexually. In plants, the end of juvenile or vegetative phase marks the beginning of reproductive phase and can be seen easily in the higher plants at the time of flowering. The flower is specialized reproductive structure of a plant in which sexual reproduction takes place. The function of a flower is to produce haploid gametes and to ensure that fertilization will take place. Typical flower consists of four whorls, viz. Calyx, corolla, androecium, and gynoecium. Sexual reproduction involves two major events, viz. Meiosis and fusion of gametes to form diploid zygote and production of genetically dissimilar offsprings. Variations are useful from the point of view of the survival and the evolution of species over the time. Sexual reproduction is characterized by fusion of male and female gametes, that is fertilization. The formation of zygote and embryogenesis, sequential events that occur in sexual reproduction, are grouped into three distinct stages, viz. Pre-fertilization, fertilization, and post-fertilization. The male reproductive wall of the flower is called the androecium, Individual members of the androecium is called stamen. Stamen consists of filament, connective, and anther, having two anther lobes. Theca. Structure of anther. Anther is generally dithecous, that is, having two lobes, and tetrasporangiate. Each lobe of anther consists of two pollen sacs. In dithecous anther, four pollen sacs are present. Therefore, it is tetrasporangiate. An immature stage of anther is represented by group of parenchymatous tissue surrounded by single layered epidermis. The heterogeneity, that is differentiation, arises when hypodermal cells get transformed into archisporial cells. TS of anther. The archisporial cells divides into an inner sporogenous cell and outer parietal cells. Sporogenous cells form sporogenous tissue. Each cell of sporogenous tissue is capable of giving rise to microspore tetrad. Parietal cells undergoes division to form anther wall layers. The wall of mature anther consists of four layers. Epidermis is the outermost protective layer made up of tabular, that is flattened cells. Endothecium is the subepidermal layer made up of radial elongated with fibrous thickenings. Inner to endothecium is middle layer made up of thin walled, that is 1 to 2 layered, which may disintegrate in mature anther. Tapetum is the innermost nutritive layer of anther wall. It immediately encloses the sporogenous tissue, that is microspore mother cell. MMC 1.3 Microsporogenesis. Each microspore mother cell divides meiotically to form tetrad of haploid microspores, pollen grains. Structure of microspore. Typical pollen grain is a non-motile, haploid, unicellular body with single nucleus. It is surrounded by two-layered wall called sporoderm. The outer layer, exine, is thick and made up of complex, non-biodegradable substance called sporopollenin. It may be smooth or with a sculptured pattern that is characteristics of the species. It is resistant to chemicals. At some places, exine is very thin, showing thin areas known as germ pores. These are meant for the growth of emerging pollen tube during germination of pollen grain. The inner wall layer, in time, consists of cellulose and pectin. Development of male gametophyte. Pollen grain marks the beginning of male gametophyte. It undergoes first mitotic division to produce bigger, naked vegetative cell and small, thin-walled generative cell. The vegetative cell is rich in food and having irregular shaped nucleus. The generative cell floats in the cytoplasm of the vegetative cell. The second mitotic division is concerned with generative cell only and gives rise to two non-motile male gametes. The mitotic division of generative cell takes place either in pollen grains or in the pollen tube. The pollen grains are shed from the anther at this two-celled stage in most of the angiosperms. Female reproductive wall of the flower is gynoecium, that is pistil. Individual member of gynoecium is called carpel, that is megasporophyll. A flower with many free carpels is called apocarpus. Example, mycelia. A syncarpous flower is one that has many carpels fused together. Example, brinjal. Typical carpel has three parts, viz. Ovary, style, and stigma. The number of ovules in the ovary varies. Example, paddy, wheat, and mango are uniovulate, whereas tomato and lady's finger are multiovulate. 1.4 Structure of anatropous ovule. Each ovule develops inside the ovary and is attached to the placenta by a small stalk called funiculus. The place of attachment of funiculus with the main body of the ovule is called hilum. In angiosperms, the most common type of ovule is anatropous, in which micropyle is directed downwards and is present adjacent to the funiculus. The ovule consists of central parenchymatous tissue, the nucellus, which is surrounded by usually by two protective coverings called integuments, with outer and inner integument. A narrow opening at the apex of the ovule is called micropyle. Chalaza is the base of the ovule directed opposite to micropyle. Embryo sac, that is female gametophyte, is oval, multicellular structure embedded in the nucellus. 1.5 Megasporogenesis. It is the process of formation of haploid megaspore from diploid megaspore mother cell MMC. Megaspore mother cell becomes distinguished in the nucellus, more or less in the center, but towards the micropylar end of the ovule. Development of female gametophyte. Megaspore mother cell undergoes meiosis to form linear tetrad of haploid cells, that is megaspores. Upper three megaspores abort and lowest one towards the center of nucellus remains functional. It acts as the first cell of female gametophyte. Generally, one megaspore towards the center is functional megaspore. It is in fact the first cell of female gametophyte. It undergoes three successive free nuclear mitotic division. Thus, total 8 nuclei are formed, 4 of which are located at each pole. One nucleus from each pole migrates towards the center and are called polar nuclei. 3 nuclei towards micropylar end constitute egg apparatus. It consists of large central haploid egg cell and two supporting haploid synergid cells. Synergid shows hair-like projections called filiform apparatus, which guide the pollen tube towards the egg. Antipodal cells are a group of 3 cells present at the chalazal end. The 2 haploid polar nuclei of the central cell fuse to form diploid secondary nucleus or definitive nucleus just prior to fertilization. This 7-celled and 8-nucleated structure is called the embryo sac. This method of embryo sac development from a single megaspore is described as monosporic development. In angiosperms, the development of female gametophyte is endospermous, that is, within the megaspore. Female gametophyte is colorless, endospermic, and is concealed in the ovule, enclosed by ovary. 1.6 Pollination. Pollen grains being non-motile, angiosperms have evolved the strategy to use abiotic agents, that is wind, water, and biotic agents, that are birds, insects, snails, to their flowers, feeding the visitors and exploiting their mobility for pollination and also seed dispersal. Pollen grains are non-motile and they are usually carried from flower to flower by means of external agents. Pollination is the transfer of pollen grains from the anther to the stigma of the flower. It is a prerequisite for fertilization because both the male and female gametes are non-motile. Moreover, gametes are produced at two different sites. Self-pollination is a type of pollination which occurs in a single flower or two flowers of a same single plant. It results in inbreeding, a selfing. In contrast, cross-pollination is the transfer of pollen grains from the anther of one flower to the stigma of another flower of different plants of same species. Pollination can be further divided into three types on the basis of source of pollination. A. Autogamy, that is self-pollination. It is a type of pollination in which bisexual flower is pollinated by its own pollen grains, offsprings genetically identical to their parents. Example: P and clitoria. B. Geitonogamy. It is the transfer of pollen grain to a stigma of different flower produced on the same plant. It is functionally similar to cross-pollination as it involves pollinating agents, but it cannot bring about genetic variations and is only of ecological significance. Example: Cucurbita maxima. It is similar to autogamy as pollen grains come from the same plant. C. Xenogamy, cross-pollination or outbreeding. It is a type of cross-pollination when pollen grains of one flower are deposited on the stigma of flower of different plants belonging to the same species. With the help of pollinating agency, it generates genetically varied offsprings. Majority of the flowering plants depend on the transfer of pollen grains. Virtually all seed plants need to be pollinated. Most of the food and fiber crops grown throughout the world depends upon pollinators for reproduction. The agents responsible for pollination have been grouped into two main categories: A. Abiotic agents, B. Biotic agents. A. Abiotic agents. These are non-living agents which include wind and water, First, pollination by wind, that is anemophily. Most of the important crops are wind pollinated. These include wheat, rice, corn, rye, barley, and oats. Palms are also wheat pollinated. Adaptations of anemophilous flowers. The flowers are small, inconspicuous, colorless, without nectar and fragrance, that is odor. The pollen grains are light in weight, dry, and produce in large numbers. To increase chance of pollination, considering vestige of pollen grains. Stigma is feathery to trap pollen grains carried by wind currents. Stamens are exerted with long filaments and versatile anthers. Stamens and stigma are exposed to air currents. Second, pollination by water. Hydrophily. Found in some 30 genera of aquatic monocots. Example, Vallisneria, Zostera, Ceratophyllum, etc. Adaptation in hydrophilous flowers. Flowers are small and inconspicuous. Perianth and other flower parts are unwettable. Pollen grains are long and unwettable due to presence of mucilage. Nectar and fragrance are lacking in flowers. Hydrophily is of two types. Hypohydrophily. Pollination occurs below the surface of the water. Here the pollen grains are heavier than water, sink down and caught by stigma of female flowers. Example in Zoostera, that is seagrass. The pollen grains are long, ribbon-like and without exine. Second, epihydrophily. The pollen grains float on the water surface and reach the stigma of the female flower, that is Vallisneria. Example, Vallisneria is a submerged dioecious freshwater aquatic plant in which female flowers reach the water surface temporarily to ensure pollination, and male flowers float on the surface of the water. Specific gravity of pollen grains is equal to that of water. That is why they float on surface of water. Some aquatic plants are anemophilous. Example, Potamogeton, Halogaris, etc. Some aquatic plants are entomophilous. Example, lotus, water hyacinth, water lily, etc.
E. Biotic agents. It includes living agents. About 80% of plants require the help of other living, moving creatures such as insects, birds, bats, snails to transfer their pollens from one flower to another. These also sustain an ecosystem and produce natural resources by helping plants to reproduce. First, pollination by insects, entomophily. It occurs in rose, jasmine, sistrum, etc. Adaptation in entomophilous flowers. They are large, showy, and often brightly colored. The flowers produce sweet odor, that is smell, and have nectar glands. The stigma is rough due to presence of hair or sticky due to mucilaginous secretion. The pollen grains are spiny and surrounded by a yellow sticky substance called pollen kit. Some plants have special adaptation for the insect visitor to help in cross pollination, that is, lever mechanism or turnpike mechanism in salvia. Second, pollination by birds, ornithophily. Only a few types of birds are specialized for pollination. They usually have small size and long beaks. Example, sunbirds and hummingbirds. Some ornithophilous plants are bombax, callistemon, that is bottlebrush, buchia, etc. Adaptation in ornithophilous flowers. Flowers are usually brightly colored, large, and showy. They secrete profuse, dilute nectar. Pollen grains are sticky and spiny. Flowers are generally without fragrance, as birds have poor sense of smell. Third, pollination by bats. Chiropterophily. Bats can transport pollen over long distance, sometimes several kilometers. Adaptation in Chiropterophilous flowers. Flowers are dull colored with strong fragrance. They secrete abundant nectar. Flowers produce large amount of edible pollen grains. Example, Anthocephalus, that is kadam tree, Ardensonia, baobab tree, or Gorakchink, Kygalia, that is sausage tree. 1.7 Outbreeding devices or contrivances. Many plants have mechanisms that discourage or prevent self-pollination to promote cross-pollination and increase genetic diversity. Plants have evolved a wide variety of sexual strategies. Genetic diversity is an essential factor for evolution. By natural selection, continued self pollination results in inbreeding depression. Thus, Plants have developed many devices to encourage cross pollination. The example of outbreeding devices are as follows. Unisexuality. In this case, the plant bears either male or female flower. It is also called dioecium. As flowers are unisexual, self-pollination is not possible. Plants may be monoecious, example maize, or dioecious, example mulberry, papaya. Dichogamy. In this device, anthers and stigma mature at different times in a bisexual flower so as to prevent self-pollination. It can be further divided into two types. First, protandry. In this type, androecium matures earlier than gynoecium, example, in disc florets of sunflower. Second, protogyny. In this type, gynoecium matures earlier than the androecium, example, gloriosa. Prepotency. Pollen grains of other flowers generate rapidly over the stigma than the pollen grains of the same flower, example apple, heterostyly or heteromorphy. In some plants like primula, primrose, there are two or three forms or types of flowers in which stigma and anthers are placed at different levels, that is heterostyly or heteroanthy. This prevents the pollens from reaching the stigma and pollinating it. In heteromorphic flower, pollen grains produced from anther pollinate stigma produced at the same level. Hercogamy. It is a mechanical device to prevent self-pollination in a bisexual flower. In plants, natural physical barrier is present between two sex organs and avoid contact of pollen with stigma of same flower. Example, Calotropis. Pentangular stigma is positioned above the level of anthers, pollinia. Self-incompatibility, self-sterility. Self-incompatibility or self-sterility. This is a genetic mechanism due to which germination of pollen on stigma of the same flower is inhibited. Example, tobacco thea. 1.8 Pollen pistil interaction. It is the interaction of pollen grains with sporophytic tissue, that is stigma. It begins with pollination and ends with fertilization. All the events from the deposition of pollen grains on the stigma to the entry of pollen tube in ovule, that is synergid, are referred as pollen pistil interaction. Pollination does not guarantee the transfer of right type of pollen. Often wrong type also land on stigma. The pistil has the ability to recognize and accept the right or compatible pollen of the same species. Thus, wrong type of pollen is discarded by pistil. Compatibility and incompatibility of the pollen pistil is determined by special proteins. This process involves pollen recognition followed by promotion or inhibition of pollen. The stigmatic surface of flower refuse other wrong type of incompatible pollen grains. A physiological mechanism operates to ensure that only intraspecific pollen germinates successfully. The compatible pollen absorbs water and nutrients from the surface of stigma, germinates and produces pollen tube. It grows through the style is determined by specific chemicals. The stigmatic surface provides the essential prerequisite for a successful germination, which are absent in the pollen. A pollen tube is finally pushed through the ovule and reaches the embryo sac. The tip of the pollen tube enters in one of the synergids and then ruptures to release the contents. Due to pollen pistil interaction, intense competition develops in the compatible pollen grains, gametes. It also plays an important role in the sexual reproduction and seed formation. Pollen grain can also be induced or germinate in a synthetic medium. Sucrose induces pollen grain germination and tube growth in vitro. Addition of boric acid facilitates and accelerates pollen germination. Artificial hybridization. It is one of the major approaches used in the crop improvement. Only the desired pollen grains are hand pollinated and used for fertilization. This is accomplished through emasculation and bagging process. 1.9 Double fertilization. Double fertilization is a complex fertilization mechanism in flowering, that is angiosperm plants. It was discovered by Navanshik in the liliaceae plants like Lilium, and Fritillaria. After a pollen grain has reached the surface of the stigma, it germinates and forms a pollen tube, which penetrates the stigma style ovary chamber and then enters ovule. The growth of pollen tube is guided by chemicals secreted by the synergids. It usually enters ovule through the micropyle. It is termed as porogamy, but in some cases it is found to enter through chalaza, known as chalazogamy, and in some plants by piercing through integuments, called mesogamy. Finally, it penetrates embryo sac of the ovule at its micropylar end. The pollen tube carrying male gametes penetrates in one of the synergids. Watery contents of synergids are absorbed by pollen tube, which then ruptures and releases the content, including the two non-motile male gametes. As non-motile male gametes are carried through hollow pollen tube, it is known as synphonogamy that ensures fertilization to take place. Syngamy and triple fusion are two events of sexual reproduction in angiospermic flowers. Syngamy is the fusion of haploid male gamete with haploid female gamete egg to produce a diploid zygote, whereas in triple fusion, second haploid male gamete fuses with diploid secondary nucleus, producing primary endosperm nucleus, that is PEN, that develops into triploid endosperm. The zygote develops into embryo. Syngamy is a type of generative fertilization, whereas triple fusion is a type of vegetative fertilization. Here, both the male gametes participate, and therefore it is described as a double fertilization. Significance of double fertilization. It is a unique feature of angiosperms. It ensures that parent plant invests a seed with a food store. Only if the egg is fertilized, the diploid zygote develops into an embryo which constantly develops into a new plant. The triploid PEN develops into nutritive endosperm tissue. It restores the diploid condition by fusion of haploid male gamete with haploid female through syngamy. It also helps to avoid polyembryony. 1.10 Development of endosperm. The triploid primary endosperm nucleus repeatedly divides mitotically to form nutritive tissue called endosperm. In post-fertilization changes within the ovule, the embryo and the endosperm are seen to develop simultaneously. The other cell of embryo sac disorganized sooner or later. The formation of triploid and endosperm nucleus triggers cell division, which leads to formation of endosperm. There are three types of endosperm on the basis of mode of development. These are: first, nuclear type; second, cellular type; third, halobial type. A. Nuclear type. It is the most common type, found in 161 angiospermic families. Here, the primary endosperm nucleus repeatedly divides mitotically without formation of without formation to produce large number of free nuclei. A big central vacuole appears in the center of the cell, pushing the nuclei towards the periphery. Later, walls develop between the nuclei. Hence, multicellular endosperm is formed. But in several cases, cell wall formation remains incomplete. Example: wheat, sunflower, and coconut. Coconut has multicellular endosperm in the outer part and free nuclear as well as vacuolated endosperm in the center. B. Cellular type. In some plants, division of triploid primary endospermic nucleus is immediately followed by wall formation, so that the endosperm is cellular right from the beginning. It is mostly observed in 72 families of dicots as in members: balsam, petunia, adoxa, etc. Halobial type. It occurs in the order Halobials of monocotyledons. In this case, first division of primary endosperm nucleus is followed by transverse fall, which divides the cell unequally. The smaller cell is called chalazal cell and larger cell is called the micropylar cell. Then the nuclei in each cell divide by free nuclear division, and then wall develops between nuclei in micropylar chamber. It is intermediate between cellular and nuclear type endosperm. Example: Asphodelus. 1.11 Development of embryo. The process of development of zygote into an embryo is called embryogenesis. The embryo is developed at the micropylar end of embryo sac. The growth of the embryo triggers only after a certain amount of endosperm is formed. After fertilization, the embryonic development begins. The zygote divides to form two-celled proembryo. The largest cell towards the micropyle is called the basal or suspensor initial cell, and smaller towards the chalaza is called the terminal or embryonal initial cell. The suspensor cell divides transversely in one plane to produce filamentous suspensor of 6 to 10 cells. The first cell of suspensor towards the micropylar end becomes swollen and functions as hostorium. The lowermost cell of suspensor is known as hypophysis. The suspensor helps in pushing the embryo in the endosperm. The embryonal initial undergoes 3 successive mitotic division to form octant. The planes of division are at right angle to each other. The lower tier of 4 cells of octant give rise to hypocotyl and radical, whereas 4 cells of upper tier form the plumule and 1 or 2 cotyledons. The hypophysis by further division gives rise to part of radicle and root cap. Subsequently, the cells in the upper tier of the octant divide in several planes so as to become heart-shaped, which then forms two lateral cotyledons and a terminal plumule. Further enlargement of hypocotyl and cotyledons result in a curvature of embryo and it appears horseshoe-shaped. The embryo development is similar in both dicots and monocots up to the octant stage. The difference appears later. In monocot embryo, single cotyledon occupies terminal position and plumule is lateral. The single shield shaped cotyledon is called scutellum. The protective sheath of plumule is called the coleoptile, and that of radicle is called coleorhiza. Finally, ovule is transformed into the seed and ovary into fruit. 1.12 Seed and fruit development. The goal of reproduction in every living organism, including plants, is to create offsprings for the next generation. One of the ways that plants can produce offsprings is by forming or making seeds. The flowers must be pollinated in order to produce seeds and fruit. Seed development is initiated by fertilization. The integuments of the fertilized ovule persist and get transformed into the seed coat of mature seed. Seeds sometimes consist of two distinct coverings, a typical outer coat, the testa, and inner thin membranous tegmen. The nucellus in the ovule may persist in some genera like black pepper and beet as a thin papery layer, the perisperm. In some seeds, the food reserves in the endosperm are partially used up in the development of an embryo. Obviously, in such seeds, the endosperm remains conspicuous and fills a greater part of the seed. Thus, the resultant seed is endospermic or albuminous. Example: castor, coconut, maize, etc. In other seeds, embryo absorbs food reserve from the endosperm completely during its development stages. Thus, endosperm disorganizes. In mature seeds, the resultant seeds is non-endospermic or ex-albuminous. Example: pea, bean, etc. The cotyledons in some non-endospermic seed acts as a food storage, and in others they are the first photosynthetic organs. Micropyle persists as a small pore in seed coat to allow the entry of water and oxygen during soaking. Fruit development is triggered by hormones produced by developing seeds. As mentioned earlier, after fertilization, the zygote is formed and the ovary begins to differentiate into the fruit, and ovary wall develops into pericarp. Pericarp is basically three-layered, which get differentiated into fleshy fruit like mango, coconut, etc. Significance of seed and fruit formation. Fruit provides nourishment to the developing seed. Fruits protect the seeds in immature condition. Seeds serve as important propagating organs or units of plants. Seeds and fruit develop special devices for the dispersal and thus help in distribution of the species. Dormancy is a temporary stage of metabolic arrest that facilitates the survival of organism during adverse environmental conditions. Structure of physiological adaptive mechanism for survival is called dormancy. Mature and viable seeds will not germinate even in the presence of favorable conditions, and they are dispersed at different places during dormancy. Viable seeds germinate only after completion of dormancy period. Apomixis. It is phenomena of formation of embryos through asexual method of reproduction without formation of gametes and the act of fertilization. Alternatively, it is unusual sexual reproduction where there is no meiosis and syngamy. Embryo develops in the ovule and ovule develops to form seed. In apomixis, when a gametophyte organ or cell produces embryo-like structure without fertilization, it is termed as apogamy. Similarly, when diploid sporophyte cell produces a diploid gametophyte without undergoing meiosis is called apospory. Example orange, mango. The main categories of apomixis are A. Recurrent apomixis. In this type, the embryo sac generally rise either from the archisporial cells or from some other part of nucellus. In diplospory, the unreduced embryo sac is derived from the diploid megaspore mother cell, example Taraxacum. In apospory, the nucellar cells give rise to apomictic embryo sac. B. Non-recurrent apomixis. In this type, megaspore mother cell undergoes usual meiotic division and a haploid embryo sac is formed. Here, the embryo sac arises either from the egg by parthenogenesis or from the other haploid cells of gametophyte through apogamy. Plants produced by this method are generally sterile and do not produce sexually. Example, Nicotiana. C. Adventive embryony. In this type, embryos may develop from somatic nucellus or integuments along with normal zygotic embryo. It is common in mango, orange, lemon, etc. It gives rise to condition called polyembryony. Genetically identical plants can be produced effectively and rapidly by apomixis. 1.14 Parthenocarpy. This term is coined by Knoll, 1902. It is the condition in which fruit is developed without the process of fertilization. It occurs naturally in some varieties of pineapple, banana, papaya, etc. In these plants, it seems that the placental tissue of the unfertilized ovary produces auxin, IAA, that is indole 3 acetic acid, which is responsible for enlargement of ovary into fruit. The fruit resembles the normally produced fruit but it is seedless. 1.15 Polyembryony. It is the development of more than one embryos inside the seed and the condition is described by polyembryony. It was first noticed by Leeuwenhoek 1719 in the seeds of citrus genus. It is the occurrence of more than one embryo in a seed which consequently results in emergence of multiple seedlings. The additional embryos result from the differentiation and development of various maternal and zygotic tissue associated with the ovule of seed. Polyembryony may be true or false depending upon whether many embryos arise in same embryo sac or in different embryo sac. In the same ovule, in addentive polyembryony, an embryo develops directly from the diploid cell of nucellus and integuments as in citrus. In cleavage polyembryony, zygote proembryo sometimes divides or cleaves into many parts of or units. Each unit then develops into an embryo. Polyembryony increases the chances of survival of the new plant. Nucellar adventive polyembryony is of great significance in horticulture.